Good afternoon, everyone. Today I'm going to share with you the research I completed this summer in the Hofstetter and Khaing labs. In particular, I was looking at the effects of glibenclamide on tissue sparing after cervical spinal cord injury in a rodent model. So to start at the very beginning, what is spinal cord injury? There we go. So spinal cord injury occurs whenever there's a trauma to the spinal cord that results in paralysis, altered or lost sensory function, or altered or lost bowel and bladder function. Cervical spinal cord injuries in particular occur in the neck, and they're the most severe type, with people with cervical spinal cord injuries often having issues moving their hands and doing really simple tasks, from feeding oneself, picking up a cup of coffee even, can be really difficult for people with cervical spinal cord injuries. As you can imagine, this is a pretty severe impact to somebody's life. And so we're looking at different sorts of neuroprotection therapies that will help people regain some sense of function after their spinal cord injuries. To do this, we looked at the effect of a novel neuroprotective agent called glibenclamide on sensory, motor, and tissue-sparing outcomes following spinal cord injury. So after a spinal cord injury occurs, there is something called secondary injury, which Surami will explain more about in her presentation. But the gist of it is the brain spinal cord blood barrier is broken, which allows for blood products to infiltrate the spinal cord. These are very toxic, and they activate immune cells that further damage healthy tissue. So the SIR1 protein channel is highly expressed in these inflammatory cells, and glibenclamide offers an FDA-approved method of reducing these sorts of inflammatory cells by preventing the SIR1 protein channel from being expressed. So we looked at glibenclamide in particular to help improve functional outcomes in cervical spinal cord injury. Of course, we didn't do this with humans because that would be totally unethical. Instead, we used Long Evans rats, and we had 3 groups. The first group was laminectomy only, second is SCI only, and the third is SCI plus glibenclamide. All 3 groups unfortunately suffered laminectomy at the C5 vertebral level. However, only the SCI and SCI plus glibenclamide groups actually had a spinal cord injury. This was considered a moderate injury. It was about 200 kilodynes of force. All of the rats survived for 8 weeks, at which point they were euthanized, and we analyzed their spinal cords, looking at a couple of key things: the amount of spared tissue, the size of the lesion that is a spinal cord injury, as well as their motor function after the injury. So this is our first big finding, right? So looking at this graph here, R means rostral or towards the nose. This is the region above the injury site. 0 indicates the injury epicenter, or the most severe aspect of the spinal cord injury, and C indicates below the injury. Glibenclamide is shown in blue, and as you can see, glibenclamide results in a lot more ventral motor neurons. Motor neurons are important because they help convey motor signals from your brain to the rest of your body. The amount of motor neurons correlate with the amount of motor function that a person has after a spinal cord injury. So we want to preserve all that we can. Glibenclamide had, on average, 16% more ventral motor neurons when compared to the spinal cord injury only group, which offers potential in humans to help raise the threshold of any sorts of motor functions that they can recover after a spinal cord injury. The second finding is that glibenclamide significantly reduces the lesion size at the injury epicenter, or the most severe part of the injury. This is super important, and as you can see, visually, they're very different, right? The amount of gray matter, which is made up of cells that help support the neurons, is vital to restoring vasculature after spinal cord injury. And basically any tissue that can be preserved, we want to preserve it. This graph is just showing you what I just said, right? So again, R is above the injury site and C is below. 0 indicates the injury site. And as you can tell, glibenclamide reduced the amount of lesion and significantly spared tissue. On average, glibenclamide had about 33% more spared tissue across the whole 7 millimeters of spinal cord that we analyzed. We did find this to be statistically significant. And this tells us that glibenclamide is really effective in sparing both white and gray matter tissue, not only at the injury epicenter, but further out away from the injury site, which is where secondary injury tends to kill tissue. Now, you might be wondering, okay, I'm glad that these rats, maybe were a little bit better than the rats with only spinal cord injuries, but why does this matter? Well, as I mentioned at the beginning of this presentation, cervical spinal cord injury is debilitating for humans, not only rats. And using rats as an animal model for humans allows increased translatability of our findings to humans. In fact, currently there is a stage 2 clinical trial going on looking at glibenclamide as a neuroprotective agent. Our findings are able to help support the use of glibenclamide as a neuroprotective agent as it relates to increasing the amount of motor neurons, decreasing lesion size, and most importantly, improving motor outcomes after cervical spinal cord injury. This final graph is showing you just how much of a difference glibenclamide makes in a rat's motor function after their injury. As you can tell, glibenclamide, the group in blue, had a statistically significant impact on motor functions, which is ultimately the most important thing for human patients. Our research shows that glibenclamide is effective in rats, and we hope that our findings will help bolster the use of glibenclamide to help people with cervical spinal cord injury be more independent, feed themselves, dress themselves, and have more motor function after their injury. I'd love to thank everyone who made this program possible, in particular, Dr. and Mrs. Ellenbogen for realizing their dream and allowing students like us to have a life-changing experience. I'd also love to thank everyone in the Hofstetter and Khaing lab for making this summer a dream for me, and it wouldn't have been the same without their support and companionship over the last 8 weeks. Thank you all so much.